Hello, welcome. We're so delighted that you'll be part of this year's summer program for the Emerge Institute. I'd just like to take a few minutes to tell you a little bit about why we formed Emerge, what exactly the Emerge Biology Integration Institute is, and then why we have a summer program and what will be covered in it. So first, a long-standing foundational interest in ecology and biology generally is how the environment shapes communities and how communities in turn shape the environment. And many of us also want to distill the lessons we learn about these interactions into improved predictions about how systems will behave through things like models. As a molecular microbial ecologist, my first lens for looking at communities is the molecules of biology's central dogma, the basic information transfer in all life. And this is where DNA is replicated to make more DNA, it's transcribed to make RNA, and translated into protein. Now, of course, cells are not alone in isolation. In communities, they are part of populations of similar cells that may have some amount of genetic variation in them, but are generally all the same lineage of organisms. And then these are also part of communities where you have a variety of different types of organisms, including phage that are present, or viruses. And these, of course, are embedded in ecosystems that have biogeochemical drivers. We have climate conditions, plants, reindeer, all kinds of things, humans. These are all part of the ecosystem. And there's interconnectivity among all of these different pieces through metabolites, shown here with these cartoon blue stick figures that can be moved between different organisms, between different categories of relationships and interactions here, imported into cells, spit back out again in new different forms, taken up by other cells, etc. They're also connected genetically, all of these different organisms, through a range of different mechanisms. And collectively, these suites of multi-scale physical, metabolic, and genetic interactions form what is called by the community genes to ecosystem scaling. And this has really been the goal of our discipline for several decades. And a lot of inroads have been made in trying to take basically lists of genes, bags of genes, if you will, and extrapolate system outputs. So this is whatever the thing is we care about the ecosystem. So it could be greenhouse gas emissions, it could be crop productivity, it could be the efficiency of our wastewater treatment plant. We have made progress in this genes to ecosystem scaling, but superb prediction has remained elusive. There's a lot of different steps going on here. Okay? And of course, the environment feeds back to genes. So this isn't a one-way path, it's a two-way path. This happens through what our institute member and my good friend Scott Zaleska and his friend Regis dubbed the 3 A's. So the first of these is acclimation, where gene expression changes in response to environmental conditions. Assembly, where the organisms present are filtered out by the environment through selection, also undergo things like drift to change the composition of the communities. And lastly, adaptation, where the gene frequencies within these communities and populations change in response to natural selection. So this side of the swoop we refer to as ecosystems to genes. And of course, when you layer on that feedback from the environment to communities, you then can have changing system outputs where the responses of the community lead to different outcomes. This is our framework within eMERGE for biological integration. It's got this somewhat unwieldy name of G2E2G, genes to ecosystems to genes. Now, of course, these interactions aren't happening usually in a steady state environment or background. These are happening in dynamically changing systems, and the preeminent consideration for probably almost any biological system we can think of these days is our changing planet, our rapidly changing planet. And so as we think about a changing environment, this may be changing system outputs from any given system we're interested in. And a subset of those system outputs can in fact feed back to the changing environment and either accelerate or dampen those changes. And this brings us to what's called sometimes the big biology problem in climate science, which is that ecosystem responses are not additive, they're emergent. And what we mean by that is that how these phages respond to increasing temperature, and this particular kind of microbe responds to increasing temperature, and this sphagnum responds to increasing temperature. You can't add up each of those separate responses and accurately predict what the system overall is going to do. And what's more, we're not just talking about temperature, we're also talking about, say, changing precipitation. And so even for one given lineage, you can't say, well, this sphagnum responds to changing precipitation this way and a changing temperature this way, and we're going to add them together and that's going to be the actual outcome. That very rarely is the case. Decades of research have demonstrated. So system responses are really emergent. And this means that even if we could predict humanity's fossil fuel burning over the coming century, we don't know how the climate as a whole, all the animals and the plants and the microbes will respond. There are these feedback loops in systems, and some unknowns in all of this are particularly consequential. There's lots and lots of unknowns here. Can we figure out which of these unknowns are consequential and put our attention there, with the definition of consequentiality being the things that are then feeding back to the system? So fundamentally, this question of G2E2G addresses twin grand challenges in biology. The first is National Science Foundation, one of National Science Foundation rules of life, which is how genotype and environment interact to produce phenotype. And the second is how we manage a rapidly changing global biosphere. Now, the G2E2G framework can be applied to really any biological system, but in the context of a changing planet, it's particularly urgent. And together, the unifying feature across these is precisely the need of biological integration to achieve predictive ecosystem understanding, or if not achieve it, at least advance it. So that covers why we forged eMERGE and why we are striving to do things in eMERGE. Now let me tell you a little bit about what eMERGE is with that kind of conceptual background. Sometimes you might hear us refer to that G2E2G thing as the fish eyeball, because it looks kind of like an eyeball, and early diagrams that we drew looked a lot more like a fish eyeball, and there was a little tail coming off the end and everything. So you may hear us through the course of the summer program and beyond refer to it as the fish eyeball. So now that you've seen the fish eyeball for this framework for biological integration, what actually is the Institute? So we applied to the National Science Foundation in their brand new Biology Integration Institute program to build on work we had previously done at this site and at others to really address this issue of emergent ecosystem response to change. And the Biology Integration Program fundamentally mandates an integration of research and education. We'll come back to that in a minute. And you can see in the background here, you've seen this slide a couple of times. This is our field site. This is a model, what we call a model ecosystem of Stordal and Meyer. So our overarching goal is to identify those emergent principles of ecosystem response to change by applying this fish eyeball G2E2G framework at a model consequential ecosystem. And to do this, we bring a lot of different disciplines together from biology and beyond. So ecology, evolution, ecosystem science, biogeochemistry, modeling, computational science, organismal biology, molecular and cell biology, social science and education, and remote sensing. And to do this, we bring a whole heap of folks from all over the world together to accomplish this. So we're roughly 90, 95 scientists at the moment from a range of institutions. And the structure of our institute is kind of like a multi-layer cake, is the way that I think about this. And so I'm going to start here. Let me show you my mouse here. Okay, so this G2E2G research activities is the thing that as scientists, we're sort of trained to focus in on. Okay, so what are your scientific questions? What are your experiments? What are your activities? And as we talked through that fish eyeball a few minutes ago, a lot of you may have been thinking about your own work and the questions that you're asking and the frameworks you're using, thinking about sort of central research activities. Okay, but there's also team science. By the time you get 90 scientists together, even if they were doing the same thing, let alone if they're working across disciplines, you really need team science support. And so there's actually a whole field that is the science of team science, studying how large multidisciplinary groups especially can work more effectively, both in industrial and academic settings. And so we were lucky enough to partner with somebody as we built our proposal, who was an expert in this, and you'll be meeting her and her team through this process. And so over the last a year and a half, roughly, that the Institute has existed, she and her team have been training us in a lot of these foundational principles and practices of effective team science. We've also got the research platform here, and this is all the tools that we use. This is our field campaign, our field sites, the samples that we've collected. We have a 10-year archive of samples. All of those things go into the research platform. Informatics and data management is a big deal in a project this size, as you can imagine. And then education and training, of course, this is the summer program is one piece of this set of activities, and then outreach, trying to connect both to the public and to other scientists. Okay, so let's see who some of the people are who are involved in this. So Jenny Cross is this amazing scientist from the Institute. She's actually the director of the Institute for Research in the Social Sciences at Colorado State University. And these are people, as we go through this, you'll see who some of the lead scientists are in each of these things. And many of them you will hear from directly through the summer program. Yes, so this is just to highlight something that Jenny will walk us through and her team will walk us through as well. In fact, we're just going to skip this one. Okay. So one of the things that we really care about, and this is somewhat separate from the team science, but kind of has arisen from it. This isn't something that was built as explicitly into the proposal, but has really emerged, if you will, from a lot of conversations. So building a safe and inclusive environment is super important. Maybe one could say now more than ever, but it's always been important, and people can't, there's a lot of vulnerability in working across disciplines. There's a lot of vulnerability with working people on the other side of the world who you may only very rarely get to interact with in person, and these days, not at all since the Institute started. And so in addition to the best practices of team science, there's also something that goes a little bit beyond that even. Although Jenny would probably, anyway, it's tied up with this, which is really forging a community that values respect and inclusivity. And so we've been doing this quite mindfully through our resources and our infrastructure. So the authorship policy and agreement to like protect people, field safety plan, again, to protect people, data sharing policies to be super clear, protect people, and our mission statement to sort of remind all of ourselves what our North Star values are as an institute. And then we have all this great team science training at the center of this that is helping us cultivate relationships, communicate clearly, manage our research more effectively, think collaboratively in new ways, informed by what Jenny's team tells us will help us be more creative. Defining what our norms and values are and thinking about what it means to lead within all these different spheres. And then we have DEI and mentor training. So you'll experience yourself several of these things, the mentor training and the navigating differences here. So collectively, and there's several other aspects too that didn't manage to get listed in this slide yet. And so Emerge's founding mission and funded mission includes broadening participation. Okay, so the next piece now is our research activities. And so the first research activity is gene expression to acclimation. So we sort of divvied up the eyeball into different pieces. So gene expression is the first direction, and then acclimation is the direction back. So for each of these, you'll see the genes to ecosystem side and the ecosystems to gene side for one piece of this. Okay, so the first research activity is led by Mike Ibba, who's a dean at Chapman University, Sarah Bagby, who's at Case Western, and Ahmed Zayed, who's here at OSU. The second research activity is all about interactions and assembly. So all the interactions are the genes to ecosystem side, and assembly is the ecosystems to gene side. And this is led by Kelly Wrighton at Colorado State University, Jessica Arnakovich at the University of New Hampshire, and Malak Tafili at the University of Arizona. I'm going to pause here for a sec because I forgot to explain this part to you, which is that our measurements and our experiments span a range of timeframes from short, medium to long. And this includes cultures, incubations, and bioreactors that are at the shorter time frame, and high resolution field observations at the decadal and multi-decadal timescales that are at the sort of longer timescales. And our research activities are fed by these different data streams. Okay, so research activity 1, which is gene expression and acclimation, is fed primarily by shorter time frame experiments, data sets. Interactions to assembly is fed by a range of time frames. This next one is genetic diversity and adaptation. So again, the two sides, the genetic diversity that's there, that is defining the genes to ecosystems interactions, and the adaptation that is defining the ecosystems back to genes interactions. And this is led by Matt Sullivan here at OSU and Jean Tyson at the Queensland University of Technology in Australia. And now we get to the funneling. So now we're trying to take, what are the most salient parts of the fish eyeball? What are the lessons we're learning? What's consequential about all of this detail that's happening under the hood of these communities? So can we funnel in the consequential details to models for improved prediction? And we use a couple of different models for this. So we use both the BioCrunch model and the Ecosys model. You'll hear mostly about these in week 2. So we'll be providing a lot of the first basic foundations of science about sort of RAs 1 through 3, and some about RA5 that you'll hear about momentarily during week 1 of the summer program. And then in week 2, we'll talk a little bit more about models. So this is run, the research activity 4, it's distilling into models, is run by Scott Zaleska, University of Arizona, Regis Ferrier from the University of Arizona, and Owen Brody from Lawrence Berkeley Labs and UC Berkeley. And then as we funnel down our insights, then the question becomes, well, we're doing this for one particular model ecosystem and really one site within that ecosystem. So how generalizable are the rules that we're identifying at this system? Are they in fact rules more broadly for systems like this? Are they rules more broadly for other kinds of systems as well? And that's the purview of RA5, Research Activity 5, that is run by eMERGE co-director Ruth Varner at UNH, Bill Riley at Berkeley Labs, and Mike Pallas at UNH. Okay, so collectively we have this awesome, amazing team of luminary scientists who are working to tackle these questions separately and in integration. Okay. And we've got the research platform, which remember is the field campaign, the informatic tools to some extent, although those come up in informatics as well, the data repository, etc. And this is run, and some of the sort of key experiments that are being tapped by a lot of the different research activities. This is run by Ruth, who you saw just a minute ago. And the last thing, Jeff Chanton from Florida State University, Rachel Wilson from FSU, and Patrick Quill from Stockholm University. Then we've got the informatics and data management side of things, which is Suzanne Hodgkins at OSU and Ben Bolduc at OSU. And I'm just pointing out the leads. Each of these activities is enacted by a whole team of people. So right now I'm just pointing out the leads for you because these are people you might have heard of in the field, and it's helpful to know, like, oh, Owen Brody, who I know from this other context, is actually involved over here. Okay, and then so that was informatics and data management, and now we've got education and training, and this is all Ruth. You will soon learn how totally phenomenal Professor Ruth Varner is. And so the pieces of this are the Summer Institute, aka Summer Program, that you are all part of. And there's an RU program, a Mentor Academy, and DEI training. And then Outreach, which is led by Ruth and Florencia von Stock at UNH. Okay, so eMERGE came to be from a suite of different programs. So Isogeny, that was funded by the Department of Energy for a decade, an REU project that was funded by National Science Foundation to Ruth for a suite of years, a NASA funded project led by Ruth, and an NSF macrosystems project led by Ruth. Okay, and I'm gonna just briefly mention the REU program because you're going to be meeting some of this year's REUers while you're at the summer program. And so Ruth led this really cool set of 4 REUs at the field site. And so this was like a 10-week summer REU. They would start in New Hampshire and then they would go out to the field at Stordal and Meyer. You can see Lake Tornatrask there in the background. They would do their own primary research out there at the site, and then they would come back and analyze things at UNH. And what was super cool about Ruth's REU program was that she would take everyone to AGU, which is the American Geophysical Union annual big meeting that happens. It's like 20,000 geophysicists and biogeochemists and stuff. It's kind of an amazing meeting. It used to be in San Francisco every year, and then it moved around. It's back in San Francisco. It's a great meeting, and she would take this entire group of REUers, and it's in December every year. She would take them in December, and so all the way through the fall, even after the REU was technically over, she would be continuing to work with all of them, along with graduate students who would help mentor them on their analyses, development, and then putting together a poster. And so they would have the whole sort of soup to nuts experience. It's very cool. And so This led to 35 different AGU presentations over those 4 years. Currently, 73% of those participants are in grad programs, 27% are in STEM internships or jobs. There's GRFPs, Fulbright Fellows, etc., and different publications that have arisen from all of this. And so, and this is actually one just from 2021 that we were so thrilled. It really came together nicely and involved several undergraduates and made it into Nature Communications. So, so regardless of what stage you're at, it's really helpful to remember, you know, some of you might have gotten into science yourselves from things like an REU experience. Everyone in the summer program has at least an undergraduate degree, and many of you have more advanced degrees as well. But it's great to have the context of remembering or being aware that undergrad researchers, not only can having that experience light a passion in them for their rest of their careers, it can really shape where they go and what they do, but it can also be a source of really powerful data that can help advance your work in ways you didn't expect and didn't plan for. And now we're starting the Emerge REU. So this was Ruth's former REU that we're talking about up here, and now we're starting the Emerge REU. This is the first year. Ruth clearly knows what she's doing with all of this, and so she and Florencia are going to be co-running this year's REU. And the reason that I'm bothering to tell you all about this is because they will be sitting in on a large portion of week 1 of the summer program materials in New Hampshire. And so they will be in New Hampshire at the same time. And we realize that a lot of the core content that we are providing to you are things that they will also benefit from. They are operating at a different level because their background is different. And so the breakout discussions, et cetera, will be separated by REU participants and summer program participants so that they can be sort of talking through things and sort of as needed going back through some of the basic material. So you will get to know this new crop of REUers in our week in New Hampshire. And I look forward to having everybody together. I think it's going to be a great experience with the tuning then of different pieces of the activities tailored to each group. Okay, so that brings us to our last program. Why a summer program? I feel like I just spoke. That brings us to our last point. Why a summer program and what will it cover? So as I mentioned, the program that NSF has that funds us in this $12.5 million research institute, and $12.5 million sounds like a lot, but by the time you have 90 scientists working across 15 institutions over 5 years, it's really not very much. But this program, but it's amazing to get all the people together, right? Like you can't do it. You cannot do this kind of big science very effectively at this scale without a big price tag. So it's amazing. And they explicitly fund research with integrated education, okay? And so that's one reason we're doing this, because we're mandated to do it. And we were actually really excited to be, because as you can see from the group's track record, this is something people care a lot about across our team. The other reason that we're doing this is because the National Science Foundation is investing hugely and focally in multidisciplinary research. So the field right now, like if you look at this year's new programs that NSF has put out, it's multidisciplinary this, interdisciplinary that, convergence this, like there is so many funding opportunities in this area. And so for all of you as fellow scientists positioning yourselves for funding, thinking about new areas of your own research, this is a growth area for funding. And so it's a good time to be preparing our fellow scientists to be able to pursue this kind of funding. Another reason is that, gosh, the reason we're doing it and the reason you all are here in part is because we recognize complex biological questions require interdisciplinarity to answer. But the flip side of that is that most of us are not well trained in doing this. You get a degree in a particular discipline with its specific jargon, its specific set of questions, its culture. And if you want to work across disciplines, you have to learn new jargon and new culture, etc. So we're not typically trained in doing this. And lastly, the need for holistic ecosystem understanding is truly urgent. This big biology problem in climate science is the, arguably, perhaps unarguably, the most urgent problem facing us as humans. And so we have species on the verge of extinction. We have social justice issues being exacerbated by the day. And we have people looking to us as grown-ups and also as scientists to try to do what we can. And we are privileged to have scientific knowledge and the ability to try to make inroads in figuring out how these systems work, because we can't manage the systems if we can't understand where the consequential levers are, right? But I am a climate microbiologist, and so for me, the climate piece of this is really pressing. I would say that many of you are not involved in climate science, and this isn't a summer program just for thinking about this in a climate science context at all. And this multidisciplinarity is key to solving a lot of questions and problems in a range of different research fields. Okay, so what will the summer program cover? Just in the home stretch here, we've got 5 core competencies that we wanted to share with folks. And so foundational science is the first one, science of team science is the second one, mentor training is the third, DEI is the fourth, and science communication is the fifth. And we'd like to get basic familiarity with each of these topics during our immersive 2-week period, and then practice and implementation of them through the following year's paper writing project. And for the foundational science, the goal is not for each participant coming from their own discipline to learn the detailed tools and analyses of the other disciplines, because that's beyond the scope of what a program like this can accomplish, and it's beyond the scope of what most of you would actually be looking for. The goal is really for the foundational science to be able to learn the fundamental concepts and terminology of one another's disciplines, and then engage robustly across disciplines in co-creation in the paper project. Okay, so for foundational science, and this is really centered around what we're trying to accomplish and emerge, so sharing with you what we are trying to do and what we have become experts in. Okay, so thinking about biomolecules for studying microbiomes, how we use isotopes. They're powerful, they're awesome, what you need to know about isotopes. Thinking about biotic and abiotic interactions in natural systems. And the sort of chemistry side of that as well, how you apply math to biological systems, how we can do biology in models, how is that represented? Thinking about remote sensing. This is a piece of it that's important in RA5, where we're thinking about scaling up to the landscape scale and beyond. And so you will get to do some drone flying, or at least some engagement with the drone. 1 or 2 drones. I'm not exactly sure how many McPalace will bring out, and you'll learn some about the sort of foundational science of remote sensing. Thinking about fundamental considerations for organizing this kind of data, and then really the ecosystems to gene side of things as well. Okay, and then for team science, thinking about how to set expectations, roles, and responsibilities in teams, how to cultivate shared understanding of language and methods. For mentor training, communicating, aligning expectations, so you can see that there's some conceptual crossover between what the data tells us is effective in team science and what the data tells us is effective in mentoring as well. And thinking about diversity, equity, and inclusivity, figuring out what you're good at as a mentor and where your areas for growth are, etc. And then a suite of things in DEI. So understanding our personal perspectives and how they inform how we interact within teams, identifying strategies for bringing inclusivity into different areas of our work, acknowledging and engaging local Indigenous stakeholders in science and what we're engaged in. And really, when we think about mentoring, bringing that lens of DEI to mentoring. And then some basic tools for science communication and approaches to storytelling. Okay, so the phases of the Summer Institute, phase 1 is preparation. That's what you're engaged in right now. So you've got about 7.5 hours of videos. This is part of that, as well as reading and self-assessment. So the 7.5 hours is all of those things. So that preparation you'll want to do over the next 2 weeks before you come. Phase 2 is our 2-week intensive program. The first week is in person in New Hampshire. And the second week is virtual, but will still be fairly full days. And then phase 3 is regular engagement over the following year to develop the manuscript and potentially to engage in additional targeted training opportunities through that time. So some of the key people, you're going to meet a host of instructors through this process, but you'll meet those the first day and through the week. And I just wanted to highlight here for you a few of the key people. So this is me, this is Ruth, go Ruth. We're the co-directors, and this is Heather Ingram. You've all been receiving emails from Heather and have interacted with her over email. She's at the University of Arizona, and she's the summer program logistics coordinator. She's about a quarter time, I think, an eMERGE coordinator. And then works in some other projects as well. But her main role and responsibility with eMERGE is to help us with the summer program. And then Dr. Brittany Fauner will also be at New Hampshire, and she is the overall eMERGE program coordinator, and she'll be helping out around the sides with different things. So you'll see her there. And so these are all faces you should look to that first day as resources to ask questions of and get help from as needed. Okay, so with that, we're so delighted that you're part of eMERGE, and we look forward to seeing you soon in New Hampshire. And don't hesitate to reach out, especially to Heather, whose email you have, and also myself about any questions you've got. Heather for all the logistics side of things, and myself for questions about the pre-work homework and stuff like that. Okay, thank you so much. Can't wait to see you soon.